Theme: Basic stages of development of human embryo. Plan: Number 1. Periodization of embryogenesis. Number 2. Fertilization and formation of zygote. 3. Cleavage. 4. Implantation and its stages. Number 5. Gastrulation and its stages. Embryonic period includes time from the moment of fertilization and formation of zygote till birth. Embryogenesis is divided into 3 periods. Primary, which includes first week of development. Embryonic, from the second week till 8th weeks of development. And fetal, from 9th week until birth. The first week of development. The first week of development is characterized by several significant events, including fertilization, cleavage of zygote, blastocyst formation, and implantation. Fertilization. Fertilization is the process by which male and female gametes fuse occurs in the ampullary region of the fallopian tube. This is the widest part of the tube and is close to the ovary. Spermatozoa may remain viable in the female reproductive tract for several days. Fertilization occurs within 12 to 24 hours after ovulation. Of the about 200 million sperm introduced into the vagina, fewer than 2 million reach the cervix of the uterus, and only about 200 reach the secondary oocyte. Spermatozoa contact with membrane of oocyte. For successful fertilization, number of sperms in 1 milliliter of ejaculate should be 60 million. During fertilization, some processes take place. These are capacitation, rotation of oocyte, acrosomal reaction, cortical reaction. Capacitation. It is the process of activation of sperms, including a series of functional changes that cause the sperm's tail to beat even more vigorously and prepare its plasma membrane. During the capacitation, a glycoprotein coat and seminal plasma proteins are removed from the plasma membrane that overlies the acrosomal region of the spermatozoa. Only capacitated sperm can pass through the corona cells. In mechanism of capacitation, significant role belongs to hormonal factors. First of all, to progesterone, hormone of corpus luteum, activating secretion of fallopian tubes. Rotation of oocyte. It is realized at the expense of beating of sperm flagella, usually 4 rotations per minute. For fertilization to occur, a sperm cell first must penetrate 2 layers: the corona, the granulosa cells that surround the secondary oocyte, and the zona pellucida, the clear glycoprotein layer between the corona radiata and the oocyte's plasma membrane. The acrosomal reaction. It is release of the contents of the acrosome. The acrosome of a sperm contains several enzymes, for example, hyaluronidase, proteases, collagenase, acrosin, and trypsin-like structures. Acrosomal enzymes and strong tail movements by the sperm help it penetrate the cells of the corona radiata and come in contact with the zona pellucida. one of the glycoproteins in the zona pellucida, called ZP3, acts as a sperm receptor. The acrosomal enzymes digest a pass through the zona pellucida as the lashing sperm tail pushes the sperm cell onward. Although many sperm bind to ZP3 molecules and undergo acrosomal reactions, only the first sperm cell to penetrate the entire zona pellucida and reach the oocyte's plasma membrane fuses with the oocyte. Fusion of the oocyte and sperm cell membranes. In the human, both the head and tail of spermatozoon enter the cytoplasm of the oocyte, but the plasma membrane is left behind on the oocyte surface. As soon as spermatozoon has entered the oocyte, the egg responds to cortical reaction. Cortical reaction. It is excretion of enzymes from cortical granules of oolemma and formation of dense fertilization membrane, which prevents entry of other sperms and prevent phenomena of polyspermy. The nucleus in the head of the sperm develops into the male pronucleus, and the nucleus of the fertilized ovum develops into the female pronucleus. After the male and female pronuclei form, they fuse, producing a single diploid nucleus that contains 23 chromosomes from each pronucleus. Thus, the fusion of the haploid pronuclei restores the diploid number of 46 chromosomes. The fertilized ovum now is called a zygote. Cleavage of the zygote. After fertilization, rapid mitotic cell divisions of the zygote, called cleavage, take place. The first division of the zygote begins about 24 hours after fertilization and is completed about 6 hours later. Each succeeding division takes slightly less time. By the second day after fertilization, the second cleavage is completed and there are 4 cells. The cleavage of human zygote is complete, uneven, asynchronous, which means that zygote is divided completely, but in different time and shape of cells is also different. The complete cleavage is caused by isoleucital type of oocyte. By the end of the third day, there are 16 cells. The progressively smaller cells produced by cleavage are called the blastomeres. Morula. Successive cleavages eventually produce a solid sphere of cells called the morula. It is formed during 50-60 hours. Blastocyst. By the end of the 4th day, the number of cells in the morula increases as it continues to move through the uterine tube toward the uterine cavity. Now it is called the blastocyst and consists of 32 cells. It is hollow vesicle filled by fluid. The blastocyst remains free within the uterine cavity for about 2 days, the stage of free blastocyst. So the stage of free blastocyst duration from 5th till 7th day. During the formation of the blastocyst, 2 distinct cell populations arise, light and dark. Light cells, from them trophoblast is formed. This represents one layer of elongated squamous cells. From dark cells, embryoblast, body of embryo, and provisory organs are formed. About 7 days after fertilization, the blastocyst attaches to the endometrium more firmly. The blastocyst eventually secretes enzymes and burrows into the endometrium and becomes surrounded by it. Implantation. Implantation is the process of penetration of embryo into the wall of uterus. It lasts during 40 hours and includes 2 stages: stage of adhesion and stage of invasion. Immediately before implantation, fertilization membrane is destroyed. Trophoblast fastens onto uterus mucosa and 2 layers are differentiated during the first stage of implantation, which is called stage of adhesion. These are the cytotrophoblast and syncytiotrophoblast. During the second stage of invasion, syncytiotrophoblast secretes enzymes that enable the blastocyst to penetrate the uterine lining by digesting and liquefying the endometrial cells. Types of nutrition. Trophoblast at first, during first 2 weeks, uses products of destruction of maternal tissues. This type of nutrition is called histiotrough type of nutrition. Further, nutrition of embryo is realized immediately from maternal blood. This type of nutrition is called hematotroph. Gastrulation. Gastrulation is the process of formation of germ layers through cell movements. The gastrulation includes 2 stages: stage of delamination and stage of immigration. First stage of gastrulation. Delamination occurs on 7th day. Cells of the embryoblast differentiate into 2 layers: external, which is called epiblast, and internal, called hypoblast. Epiblast includes material of ectoderm, mesoderm, and cord. Hypoblast includes material of endoderm. Cells of the hypoblast and epiblast together form a flat disc referred to as the bilaminar embryonic disc. Soon a small cavity appears within the epiblast and eventually enlarges to form the amniotic cavity. The second stage of gastrulation. It is called immigration. Immigration starts on 14th-15th day. It involves the rearrangement and migration of cells from the epiblast. As a result, the primitive strake is formed, a faint groove on the dorsal surface of the epiblast that elongates from the posterior to the anterior part of the embryo. The primitive strake clearly establishes the head and tail ends of the embryo, as well as its right and left sides. At the head end of the primitive strake, a small group of epiblastic cells forms around its structure called the primitive node. It is source of head part, giving rise to cord. The cord is base of axial skeleton. Further from it, vertebra forms. Cells of the epiblast move inward below the primitive strake, and some of them displace the hypoblast, forming the endoderm. Other cells remain between the epiblast and newly formed endoderm to form the mesoderm. Cells remaining in the epiblast then form the ectoderm. So, during the second stage of gastrulation, the bilaminar or a 2-layered embryonic disc consisting of epiblast and hypoblast transforms into a trilaminar embryonic disc consisting of 3 primary germ layers: the ectoderm, mesoderm, and endoderm. The primary germ layers are the major embryonic tissues from which the various tissues and organs of the body develop. Thank you for attention.